Hello everyone, welcome to the new episode of Micro Talks. Today we will be discussing the Hershey and Chase experiment. In the year 1953, Alfred Hershey and Martha Chase, they published a paper stating that DNA was the genetic material. This study confirmed the results that were put forth by Griffith in the year 1928 when he carried out the Griffith transformation experiment using mice and S. pneumoniae. Later on, also, the same hypothesis was proved by a very MacLeod in 1930s and 1940s, stating that DNA was indeed the genetic material. Let us have a look at the hypothesis that was put forth by Hershey and Chase. So the working hypothesis that they put forth was that DNA is the genetic material. Their experimental model was a bacteriophage called T2. As we all know, bacteriophages are viruses that attack bacteria, and like all viruses, phages also need a live cell to reproduce. They infect the live cell and they complete the lytic cycle to release progeny virions. Hershey and Chase knew that the phage T2 contains only DNA and protein. The natural host for the phage T2 is E. coli, and this releases progeny virions after completing the lytic cycle. The structure of this icosahedral phage T2 is very simple. There is an icosahedral capsid that contains the genetic material, and upon adsorption onto the cell surface of E. coli, the phage injects the genetic material inside the bacterium and it leaves behind the empty capsid on the cell surface, or the empty shell on the cell surface, which is known as the phage ghost. So this will not contain, the phage ghost will not contain any genetic material, but it will be made up of all the structural proteins that have been coded for by the phage. Let us look at the experiment that was conducted by Hershey and Chase. So they conducted this experiment in two parts. In part A, they prepared the radioactively labeled T2 bacteriophage. So E. coli, when grown in a medium that contains radio-labeled isotopes of phosphorus and sulfur. Now we know that DNA contains phosphorus and no sulfur, and proteins contain sulfur and no phosphorus. The radio-labeled isotope of phosphorus is 32P and that of sulfur is 35S. The E. coli takes up whichever isotope is provided to it in the medium, and the E. coli will then incorporate either the labeled phosphorus in the DNA or the labeled sulfur in the proteins. Now when the phage has infected the host and has entered into the lytic cycle, the phage will utilize the entire host cell machinery to complete the lytic cycle and make the progeny phages. So in part A of the experiment, this was again conducted in two steps. In the first step, they took T2 phage and mixed it with E. coli. So the T2 phage was allowed to infect the E. coli, and this was then grown in a medium containing radio-labeled phosphorus. After the cell lysis, it was found that the progeny phages were all labeled with the DNA that the progeny phages contained, all of them had radio-labeled phosphorus. In the second step of this experiment, The T2 phage was mixed with E. coli and it was grown in a medium that contained radiolabeled sulfur. Infection was allowed to proceed and the host cell lysed, and the progeny phages that were obtained, they all contained the proteins which had the radiolabeled sulfur in them. So in this part A of the experiment, what Hershey and Chase did was that they prepared radioactively labeled T2 bacteriophage. One part of the bacteriophages had the DNA which was radioactively labeled, and the other part of the bacteriophages had the proteins which was radiolabeled. Now coming to the part B of the experiment, which proved that DNA is the genetic material. The progeny phages that contained radiolabeled DNA, which were obtained in part A, they were mixed with E. coli and they were allowed to adsorb onto the cell surface. Thereafter, that entire mixture was put in a blender and observed for the radioactivity. So it was found that the radioactivity was recovered in E. coli and empty phage hosts were observed. So empty phage hosts were observed, which meant that the DNA that was present in the progeny phages, the radio-labeled DNA which was present in the progeny phages, was all transferred to the E. coli host. Mind you, here the E. coli was not grown on any medium containing any isotopes. So therefore, the radioactivity was passed on to the phage progeny. The labeled phage DNA had entered the cell as part of the lytic cycle and the radioactivity was seen in the host cell. And upon completion of the lytic cycle, all the progeny phages had radiolabeled phosphorus in the DNA. And the phage ghosts which were on the cell surface did not show any radioactivity. In part 2 of the same experiment, the progeny phages that contained radiolabeled protein that was labeled with 35S. This was obtained in the previous part A of the experiment. So these progeny phages which were obtained in part A of the experiment, which contained radiolabeled sulfur, were mixed with E. coli. Again, the phages were allowed to adsorb onto the cell surface, put in a blender and checked for the radioactivity. So radioactivity in this case was now found in the phage host which was there on the cell surface, but it was not found in the E. coli host. And the progeny phages also were not found to be radioactive. So the infecting phage contained the labeled protein and did not contain the labeled DNA. So upon injection of the genetic material during the lytic cycle, normal DNA entered the host. And obviously the labeled protein did not. It remained on the phage hosts in the surface of the cell. And since normal DNA had entered the host cell, upon completion of the lytic cycle, normal progeny phages were produced. And there were no radioactivity that was found in the normal progeny phages. Radioactivity was only found in the phage host which was left behind on the cell surface. So in this particular slide, I have taken the diagram from the iGenetics by Peter J. Russell, which explains this entire experiment diagrammatically using certain illustrations. And as you can see and have understood from whatever I have explained, Hershey and Chase therefore reasoned that because it was DNA and not protein entering the cell, DNA must be responsible for the function and reproduction of phage T2, thus proving that DNA is indeed the genetic material of phage T2. We know that only genetic material is passed from parent to progeny during the lytic cycle and the phage reproduction. Protein is not passed. Hence, the progeny did not show presence of any labeled protein. So this amply proved the fact that DNA was the genetic material in phage T2. Alfred Hershey won the Nobel Prize in 1969, which was shared. He won it in the area of physiology or medicine for their work titled Discoveries Concerning the Genetic Structure of Viruses. In this particular video, I have used iGenetics by Peter Russell as a reference. I really do hope that you have understood today's topic. Thanks for watching my channel, and I do hope that all my viewers have found the content useful and easy to understand. Do like, share and subscribe to my channel. Thank you for watching patiently.